Can I?
Yeah.
Alright, so I'm just gonna begin.
Isabel, this is for you.
There's only one man. I thought Samantha.
Oh, Rachel too.
Oh, Rachel. Anyway, okay, now for this topic, I'm gonna tell you that practice makes perfect. Okay, if you are lazy, you copy your friend's work, you're gonna forever struggle with this topic. Okay, so I recommend that you try as many questions as you can, including the supplementary questions at the back. It's actually not a lot. Okay, so if you look at the supplementary questions, I don't know where they start from. Let me see. Where is it? Page. Can somebody find the supplementary one? Take what?
Page.
Nice. Are they here? Page 34. Okay, now if you look at the additional practice questions, those are supplementary. It will not be covered during tutorial, but I may do it during bioclinic. Okay, whenever. So anyway, no matter what, please practice. Okay, practice. The more you practice, the better you are. The tutorial question is so minimal. It's like one of each type of inheritance. That's not enough. Okay, so please try it. And another thing, like what your lecturer said, it is very important for you to read the question carefully. Okay, because all the clues are given in the question. Alright, so if you miss out one word, that's it already. Okay, now this is basically, this page here is the summary of your inheritance already. Okay, so how do you approach genetic questions? There are some questions to ask yourself. If you look at the question, you need to ask yourself, how many characteristics are being investigated? Characteristics and traits, we use it kind of interchangeably. So if let's say we are looking at this question, there's only color of seed. That's just one trait. Okay, if you have color of seed and shape of seed, that's two traits. Okay, so it could be one trait, it could be two traits. Okay, so in your syllabus, usually one or two. Okay, so I mean, if you do more questions, maybe there's three and whatever. Okay, now this question number 2 is really important. Please ask yourself how many genes are involved, okay? Because you learned many different types of inheritance patterns, right? So this topic is like you're a detective like that. You're trying to find out what is the inheritance pattern. So over here, if you're looking at one gene that's controlling one characteristic, what is the inheritance? What is it called? One gene controlling one characteristic is called monohybrid inheritance. If it is two genes controlling two characteristics, dihybrid inheritance. If it is two genes controlling one characteristic, it is nice. Hello. Look at point number 7 in your worksheet. Point number 7, what is it? Hey, epistasis. Yes, who is that? Jingxian. Epistasis. Okay, gene interaction. Okay, so it could be one characteristic that's controlled by two genes. Okay, it's just that they interact in some way. Maybe it inhibits, or I don't know how. It complements each other, things like that. There's one more that's not here. Can you just add it in? It could also be many genes controlling one characteristic. Now this one you will learn in the next lecture slot. So that will be, is it tomorrow? No, Wednesday's lecture. So many genes controlling one characteristic, we call it polygenic inheritance. So that's the last part of your lecture already, like polygenic inheritance. Okay? Now, question number 3. For every gene that you are looking at, how many alleles are involved? Most of the time, how many alleles do you see? 2. Okay, so most of the time, it is 2 alleles. Okay, but in very few situations, you can see more than 2 alleles. We call them multiple alleles question. Example of multiple alleles?
Rooster.
Oh no, that's not multiple alleles. Good try. Something you learned in secondary school. Blood, yeah, somebody said blood type. Right? controlling your blood type, you have 3 alleles for that gene that's controlling blood type. So they are considered multiple alleles already because it's more than 2. Okay? Now, the alleles of a gene have different relationships. They could be in a dominant and recessive relationship, so we call them complete dominance. Complete dominance means in the allele pair, there is one that is dominant over the other, that's recessive. Okay? Now, also, there is what we call co-dominance. There is also what we call incomplete dominance. Now, of course, co-dominance and incomplete dominance is different. We will look at some questions and you will be able to see the difference. Now, the next question to ask yourself, point number 5. Where are the genes located? Is it on the autosomes? What is autosomes? Non-sex chromosome. So basically, in humans, we have 23 pairs of chromosomes. Which chromosome number is your autosome? 22, yes. So actually 1 to 22, all of them are autosomes. The 23rd pair of chromosomes in humans, we call them sex chromosome. They are either XX or XY, depending on your gender. Okay, now the next one, of course the genes, if it's located on autosomes or sex linked, there's an effect. Okay, we'll look at that. Now, the genes, if you're looking at more than one gene, it could be located on the same chromosome. Now, if the genes are located on the same chromosome, we call them linked genes. Right? Why are they linked genes? Because the genes tend to be inherited together. Okay? Now, if we are looking at more than one gene and they are on different chromosomes, we call them unlinked genes. Okay, so that's what the terms mean. The next one. This part is very important. Can you highlight? Okay, now over here we have something called the Mendelian ratio. Okay, now who is Mendel? Okay, yeah, the monk, the priest or monk or I don't know. Gregor Mendel, right? So basically he's called the father of genetics. He did research on pea plants, right? And basically he came up with all these Mendelian ratios. So if it is you're looking at monohybrid inheritance, meaning that you only have a single gene locus, right? According to Mendelian laws, right, if you cross two heterozygous parent plants together, you're gonna get an offspring ratio of 3:1, right? If you cross a heterozygote parent with a homorecessive, you're gonna expect a 1:1 offspring ratio. Please memorize this, okay? Well, if you don't need to memorize, if you do enough, you'll remember somehow, okay? Now, Mendel also discovered if you are looking at dihybrid inheritance, if you cross two double heterozygotes means at each gene locus is heterozygous. Okay? So if you cross two heterozygote parents together, you're going to expect a 9:3:1 offspring ratio. If you cross a heterozygote with a homorecessive, you're going to get 1:1:1 ratio. Okay? Now, sometimes, okay, so this is what you expect when you cross all of these things together, okay, all these ratios. You are always expecting such ratios. But sometimes, you don't see such ratios. How come? In what situations do you get non-Mendelian ratios? Example, situations of non-Mendelian. When is it that when you cross two heterozygote individuals together, you don't get a 3:1? Good, incomplete dominance, right? So, example, if you have incomplete dominance or a co-dominance, if you cross two heterozygote, instead of getting 3:1, you're gonna get is a 1:2:1 ratio. Okay, you're gonna see more of these questions later. In other examples, any other examples besides co-dominance, incomplete dominance, you're not going to see this ratio. Any other examples? Yes? Any other examples? Huh? No. Random fertilization will still get this ratio. Guys, are you okay now? You should be throwing out examples freely. Hello? Point number 7. Can you see point 7? What is it? Epistasis. By the way, if you have not realized epistasis ratios, all of these epistasis ratios is actually what cross with what? To get all these epistasis ratios. Yes, you're crossing two double heterozygous parents together, and what you are expecting is I am supposed to get 9331. However, because of epistasis, I don't get 9331. I get a modified 9331. So what is the modified ratios? These are the modified ratios. Right? It's because the two genes interact in some way, either inhibit or they complement each other. Because of that, I get modified ratios. You see what I mean? Okay, so these are non-Mendelian as well. Any other examples of non-Mendelian? Look at everything that we have said so far. What other things do you expect non-Mendelian? That means you cannot have expected ratios of 3, 1, 1, 1, 1. Huh? Yes? Polygenic inheritance, okay. Polygenic, yes? Sex chromosome, very good. If you have sex linkage, you don't get expected ratio basically of all of these. Any other one? Last one. There's still one more. Autosomal linkage. Right? If you are looking at linked genes question, you're not gonna get all of these ratios that you expect. You're gonna get more offspring that have the parental genotype. I mean, if you know what I'm talking about, okay? So any of this, let me see, did I cover everything? Yeah. Now, if you are Mendelian inheritance pattern, right? What are the laws that Mendel came up with? Okay, law of segregation, yes. So what does that mean? Okay, what's the second law? There is law of segregation and the next one is law of independent assortment. Okay, we talk about law of segregation first. Alright, the reason why Mendel have all these ratios is because of law of segregation. So what does that mean? What is law of segregation? Okay, during anaphase, what is being separated? Okay, law of segregation is not talking about homologous. Not really. So you're going to say, for law of segregation, each gamete receives only one of an allelic pair. Okay, so if you have big A and small a, right, you only have one. You cannot have both. Example, if you have this parent, right, what is the possible gamete that they can form? Big A and small a. You will never ever inherit both alleles. Okay, you only get one allele of every pair, allelic pair. Okay, that's law of segregation. And what is law of independent assortment? You learned it in cell cycle. Which phase is relevant? Your law of independent assortment. Anaphase, okay, more importantly, metaphase 1 and anaphase 1. Okay, so let's recap law of independent assortment here. Okay, so what is independent assortment talking about? At metaphase 1, you have two rows of homologous chromosomes joining. Homologous chromosomes have already paired up at prophase. They are aligning in the equator in two rows. Right? Now, when the way they arrange, there's two possibilities. It could be that the paternal one all on the left. It could be the paternal one left, one right. You get what I mean? And because of the independent assortment, the way that they arrange can be random, you're going to get 4 different types of gametes that form at the end. Okay, you can see these 2 gametes are only having the paternal one alleles. These 2 gametes only the paternal one, whereas these 4 gametes get a mixture. Alright, so this is what I mean by law of independent assortment. Okay, now this is only possible, law of independent assortment is only possible if you have genes that are located on different chromosomes. Example, if let's say we are talking about one gene here, gene A here, and gene B here, right? You can actually so-called separate them. They won't be inherited together because they are on different chromosomes. Okay, I hope you know what I'm talking about. Anyway, where was I? What else? Did I cover everything? Okay, now the last thing is this. Okay, now this tutorial, besides epistasis, what else are we going to do? We are going to do pedigree analysis. Okay, yes, I don't know why you all don't know what pedigree means. You look at question number 5, right from the back. Right from the back, additional question, that is a pedigree. Question 5, do you see? The boxes and the circles, the square and the circle. Question 5. Question 5. Right from the back, yes. The one, the picture, a big gross one. Question 5 from the back. Here. Wait, take out the prep now. Okay, all of this is your pedigree analysis, so I hope you can analyze a pedigree chart. Okay, that is actually takes some skill. And of course, lastly, what you have just learned today, chi-square test, statistics. Okay, so these are all part of your inheritance topic. Any questions? Of course, we'll be doing each of them slowly. If no questions, can we move on to the tutorial? So, tutorial. Now, look at the LOs. Can you highlight LOA? Just put a star. Now, contrary to students' belief, okay, you can be asked definitions in bio. Okay, so the most commonly asked definitions for Cambridge exam is in genetics topic. They may ask you all of these terms. So make sure you know how to describe all of these terms. If you cannot describe, you go and memorize, okay? Alright, now the first part, okay, you're going to see question number 1 is a definition type of question. So let's allocate the marks, okay? Very easy to allocate 1 mark for gene, 2 marks for allele, maybe 2 marks for gene locus. How do I know how much marks to allocate? Depends on what? Number of lines, exactly. So let's come up with a definition for gene. Very easy, okay, just copy only. So what is the definition of gene? Yeah, okay, I don't think you have a problem. So basically, it's a very specific sequence of nucleotides. Okay, and this sequence actually codes for either RNA or a polypeptide. Please, if you have done it, can you please underline the keywords? Alright? Shouldn't have a problem. Now, how about alleles? Very good. This one, no issue also. Alternative form of a gene shouldn't be a problem. The problem is with elaboration. Okay? Okay then, so let's talk about alleles, right? The word gene represents a sequence of nucleotides that code for RNA or polypeptide, right? So in this case, it could be an enzyme that synthesizes a pigment that gives rise to eye color. Alright, now the term allele is an alternative form of a gene. So over here, this gene, right, it could be 2 alleles, dominant allele A, recessive allele small a. Okay, now let's say, for example, this is homologous chromosome. Right? This is the paternal one, this is the maternal one. Now, how do you elaborate the term alleles? What can you tell me? Alleles? You want to elaborate to get more marks, right? What can you say? Yes? Alleles? Same position, yes. Please use keyword. They occupy the same gene locus. Okay, so some of the things that you can say. Over here, you can see that, let's say the paternal chromosome, big allele A, maternal one, small allele A. They occupy the same position. Okay, the same gene locus. On a pair of homologous chromosomes. Other than that, now what is this point talking about? Right, you can have multiple alleles, like blood type. You can have IA, IB, and IO, right? You can have many types of alleles in the human population. But in a diploid individual, in your cells, you can only have a maximum of 2 alleles. Right? So you're either IA or IAIO. You get what I mean? You cannot be having 3 alleles. Alright, so a diploid individual can only have 2 alleles even if you have multiple allelic form. And of course, only one of each pair is represented in the gamete due to law of segregation. Now, how do you define gene locus? This one should be quite easy. Gene locus, it is a gene locus. Then it's a position. Very good. It is a position of occupied by a gene on the chromosome. By the way, can somebody take photos and send to the group chat? Wait, let me zoom out a bit. Can? Okay, I zoom in. Okay, finish. So slow one. Cannot. It's all from the lecture notes. It's not difficult, right? Okay, I'm moving on. You okay? Can Okay, moving on. Now I have a homologous chromosome pair here and also here. They are the same. What is the difference in my drawing? They are representing the same pair of homologous chromosome, but sometimes when you look at questions, it can be drawn like this or it can be drawn like this. What is the difference? On top versus at the bottom. Got difference or not? Of course got difference. What's the difference? Yes, over here you see sister chromatids. So that means what has happened? Replication. Guys, this is all basic questions. You shouldn't have a problem. Over here, this is your DNA molecule that has not replicated. This one is after replication, so sister chromatids are formed. Okay? Now, if I want to label my sister chromatids, okay? Over here, big allele, A. Over here, big A. Why not small a? A lot of you like to label as small a. These is big A because genetically identical sister chromatids. Over here, this one, small a, this one, small a. Over here, I have another gene locus that I'm considering. Let's say gene B for hair color. This one, big B, big B because genetically identical. Small b, small b. Okay, so this is basically how homologous chromosomes can be represented. Now, the next question that I will be looking at is, how can genetic material be exchanged between homologous chromosomes during meiosis? So this is my homologous chromosome. During meiosis, how can I exchange genetic material? Crossing over. Now, this is 5 marks. So you're going to break up crossing over into 5 steps to give me 5 marks. Step number 1, what must you say? Prophase 1, you need to tell me phase. By the way, cell cycle phases are keywords. So prophase 1 is a keyword. You need to tell me crossing over between homologous chromosome during prophase 1. Now, how does crossing over happen? Over here. How does crossing over happen? What exactly is happening? Can you describe in layman terms? What happens?
They pair up.
Okay, yes, there is pairing first during prophase 1 and then the pairing is called what? Synapsis. Good. After pairing up, what happens? Chiasma is formed, then what happens?
Break and rejoin.
What break? Phosphodiester bond break, yes. So basically what you're going to have is you're going to have a segment of chromosome that is exchanged. Okay, now can you label the alleles for me again? On top, what do I have?
Big A, small a, small a.
Bottom?
Big B, small b, big B, small b.
Right, so basically what has happened during crossing over, there is a breakage of phosphodiester bond leading to a segment of chromosome to be kind of exchanged between the non-sister chromatids. Right? Now, what is the result? This is so important, right? Crossing over. How come? Because it's going to affect what gametes I get at the end. Now, one of the gametes, okay, during meiosis one separates what? Homologous chromosome. So, meiosis 1 separates homologous chromosome. Meiosis 2 separates what? Sister chromatids. Okay, but they are non-identical now. So because of that, one gamete is going to get this chromatid, one is going to get this one, the third gamete is going to get this, fourth gamete is going to get this. Because of that, I am going to get 4 different gametes, and what do I have inside? This one will be allele big A, big B. The second one, A, B, A, B. Now, these genes, they have a relationship. What do you describe their relationship? They are linked. Can you see they are different genes on the same chromosome? So they are linked and they tend to be inherited together. Now there are names given to this type of gamete. This type of gamete, this one and this one, what are they called? Gametes. What gametes are they? Homozygous, what are you talking about? These are called parental gametes. This is your autosomal linkage question already. Parental gametes because it's exactly the same as the parent, right? Big A linked to big B, big A linked to big B, small a linked to small b, right? Same as the parents. Whereas these two, they are called
Recombinant.
very good recombinant gametes. Why? Because it has been affected by crossing over such that your big A is no longer linked to big B, it's now linked to small b. Okay, so crossing over will change, okay, the inheritance of your linked genes, basically. Okay, so how do you describe the result? The result is crossing over will change the combination of alleles on the chromosome. I repeat, what is the effect of crossing over? It changes the combination of alleles on the chromosome. Right, so instead of big A with big B all the time, I can have big A with small b now. Okay, so whatever I said can be summarized in here. So can you take a photo? Okay, so make sure you have all these keywords. Can you take a photo of the drawings also? Send to the group chat, thanks. When are they back?
Never.
2 weeks. I think one of them will be back tomorrow. I think. Is it Isabel?
Yes.
Tomorrow, I think. Plus the Friday, luckily our class not affected. But I don't know that anyway. Okay, can you please make sure you underline all the keywords? Okay, so look at the last point. The last point is very important. Basically, it shows you the consequence of crossing over, which is changing the combination of alleles on the chromosome. Is the video okay? Ken, is it still recording? Yeah. This part, then I got picture. Okay? Okay, due to this virus, I don't know what other measures are going to come up. You all got to learn independently, okay? Everything has to be online and everything, so count on yourself. If you slack off, too bad. I know of some students, right, their school did online lecture, and the school teachers actually, that means they have no lectures at all, it's online. So the school teachers will, yes, exactly what I mean. School teachers will track, whether they have gone through the lecture. So they will track whether they watch the lecture. You know what some students do, I heard? They play the video and then they go and play basketball. Hello, this is called 骗自己. You know what's 骗自己? In Chinese means you lie to yourself. Why you do that? So stupid, right? You satisfy the teacher only, but then you lose out, right? And Anyway, can I move on? Okay, can I move on already? Okay, Anybody needs more time? By the way, please go and do your tutorial questions up to question 9 because the lecturer will cover 8 and 9. I'm trying to cover as much as I can so that you are clearer before your lecture when they do tutorial question. You know what I mean? Okay, so I'm sorry I got to move on. Can you copy from the online answers? Okay, just refer to the WhatsApp. Okay, now the next question here. Alright, you have a diploid cell. Now this cell has to be a germ cell. How do you know it's a germ cell? Okay, it is 2N, yes. Besides that,
You give off gametes.
exactly, because they form gametes, it has to be germ cells, such as a cell from your ovary or cell from your testis, right? Now, this diploid cell is 2N. 2N means what? I mean, it means diploid, yes, but what does that mean actually? 2 sets of chromosomes. So, how many sets do I have? 1 set from Papa, 1 set from Mama. Okay, so that's 1 set. Now, which one is homologous? I hope you can recognize. These two are homologous to each other. Now, how do you define homologous chromosome?
Look the same.
Look the same. Can you use proper language? Same length. Yes, same length, or you can say same size.
Same gene locus.
Same centromere position. Yes, same gene locus. So they are homologous chromosomes, right? Alright, now, so we have a diploid cell and we want to form gametes, which is haploid. 1 set of chromosome only. So in each of the gamete, how am I going to draw? I'm supposed to get only 1 set. So what am I going to draw inside my gametes? Some of you, very good, only 1 long and 1 short. Okay, so I'm going to get 1 long and 1 short. Can I change the screen?
Yes.
Okay, I change already, So can you see that you should be getting all of these, 1 long and 1 short? Okay, now based on this, if you have a cell that is like this, okay, undergoing meiosis, I'm going to get 4 possible gametes formed like this. Okay, so this is the reason why. Now how come, okay, I can get 4 different types of gametes? Why? What is the, no, it's nothing to do with crossing over. I don't know why everybody says that. It's nothing to do with crossing over, you know. How come you get these 4 gametes? Yes, it's due to independent assortment and segregation. Right, the reason why you can form 4 different types of gametes from this cell is due to independent assortment and segregation, i.e. Mendel's laws. Okay? By the way, how do you describe this individual? Let's say this guy here. Oops. This guy, his germ cell here. How do you describe this individual? He is? Yes, heterozygous. Is that all you're going to say? At genes R and Y. So basically you can say this guy, he is heterozygous at both gene loci. Right? The gene locus for R and gene locus for Y, he is heterozygous. Could it be heterozygous for R and homozygous for Y? Can. Why cannot? It could be also. So if you're looking at different gene locus, you need to be very clear in your answer. Okay, when you use the term heterozygous, homozygous dominant, homozygous recessive, make sure you tell the examiner which gene locus you're describing. Okay, because the individual can have different genotype. Okay, so the next question is basically asking you, explain your answer to your drawing based on your knowledge of Mendel's law. So what do you think you need to write in your answer? To explain, okay, how come I get all of these gametes or all of these combinations of chromosomes? What do you need to cite? It is how many marks? Huh? 3 marks. So 1 mark for? 1 mark for?
Which phase?
Oh, which phase? Okay, which phase? Anything else? This is Mendel's laws. Mendel's laws.
Both of his law.
Both of his laws, very good. Law of segregation and law of independent assortment. By the way, the genes that I just drew, gene R and gene Y, are they linked genes or unlinked genes?
Unlinked.
Unlinked. How come they are unlinked?
Not on the same chromosome.
Very good. They are on different chromosomes. Okay, so they are not linked. You took a photo already, the eraser. Next question, describe 3 possible sources of variation as a result of meiosis. I mean, this one ask until don't want to ask. I'm sure. I hope you can answer part C. Do you know how to do it? 3 sources of variation. First source of variation. Is that all you're going to write? Yes, prophase 1. You have to tell me it's during prophase 1, crossing over. Source of variation number 2. Independent and assortment of homologous chromosomes during metaphase 1 and anaphase 1. How about number 3? Yes, I'll talk about fertilization later. It's rejected. Okay, yes, I'm sure you got all the marks. 3 marks, ask until don't want to ask. If you did not get any of the keywords, please go back and revise. This is asked in cell cycle before many times also. Now, some students, I wonder who, is going to write random fertilization, random fusion. It will be rejected. Why? Some students will surely write. How come it's rejected? Yes, reject random fertilization. Open your eyes and see. Okay? Random fertilization is not part of the meiosis process, so it will be rejected. Only these 3 processes are part of meiosis. And of course, last question. You have an albino person. Okay, never mind, I'll give you a blush. Is it okay?
Yes.
Now you have an albino person, they say albinism is controlled by a recessive allele. So what does that mean?
You must get both recessive.
2 recessive alleles in order to be an albino person. So let's say this person is a homozygous recessive, small r, small r. Now if you look at this person's chromosomes during telophase, allele small r do you expect? Okay, let's label the alleles first. Let's say the paternal allele, what should I label it as? Big R or small r? Small r lah, the person doesn't have big R, right? And over here, maternal one? Also small r, okay? And at telophase 1, what is happening at telophase 1? Telophase 1, separation of homologous chromosome 1 is being dragged to each pole. Based on that, your answer is? I mean, I'm doing part D already. Answer is 2, yes, answer is 2. Right, because at one pole, I'm going to have 2 alleles, and another pole, another 2 alleles. Okay, so we are going to stop here. Can you do your write down? I'm still got. Okay, so yeah, see you for. We still got time, eh? Okay, we do one last one then, since you don't need to write down. Can I do the next one? Just the MCQ very quickly. Okay, so the MCQ is quite simple. Basically, you have a gene, okay? The gene with 2 alleles, allele this one and allele this one. This one is a mutant enzyme made, and this one is a functional enzyme, giving rise to green pigment. Okay, this one gives rise to no pigment. Okay, so one gene, 2 alleles, one gives rise to functional enzyme, one codes for a mutant enzyme, no pigment versus green pigment. Now, if you cross 2 heterozygotes together, right, how come you will get this sort of ratio? So let's say, for example, 2 heterozygotes cross together. What do you expect to get? You're supposed to get this, and this. Okay, how come I don't get a 1-to-1 ratio? I mean, how come I get a 2:1 ratio? Why?
One is dominant, one no.
Can we look at the likely explanation? Part A, CG is dominant. This one not so sure, but definitely CW is not dominant. Now, there definitely, okay, can we try and match to the phenotype? I'm supposed to get 1 dark green and 2 pale green. So which one is likely to be the dark green? 1, 2, 3, 4. Which one? 1, 2, 3, 4. Definitely the one, because I have presence of more alleles that can make the green pigment. So this one has to be dark green. Okay, this one is likely to be pale green. And the last one, how come I don't see any third phenotype?
Why?
Because it is probably dead. So we call this a lethal genotype, and D is the answer. Okay, so that's all for today. We will continue tomorrow. Can you please do more questions? Oh yeah, I will talk first.
Okay, try 4.
Bye, Belle.
Rachel.
Bye, Rachel.
Bye, Rachel.
Bye, Cal.
Don't know if you're coming back, but bye.